Immunology Part 3. In the Part 1, we learned about the major histocompatible complex molecules, and in the Part 2, learned about cell-mediated immunity. Now we are moving towards the humoral immunity. Humoral immunity, which is possible by B lymphocytes. Let me once remember that MHC molecules, how they are participating here. We learned earlier, like major histocompatible complex molecules are of two types, MHC1 and MHC2. As there is no matter of MHC1 at present, because now we are discussing about the humoral immunity, whereas MHC1 molecules are going to participate only in the cell-mediated immunity. So now we are discussing about humoral immunity. That means let's focus on MHC class 2 molecules. Learned earlier in the part 1 about the MHC class 2 molecules. Let's see once again. MHC class 2 molecules are present in antigen presenting cells like macrophages, dendritic cells and B lymphocytes. B lymphocytes also act as antigen presenting cell. Then they submit the antigen to the T helper cells. They submit the antigen to the T helper cells. When it was given to the T helper cells, there are two possibilities. They may give it to the T cytotoxic cells for the cell-mediated immunity that we are discussed in the part 2. And now we are moving towards the T helper cells. If they submit to the B lymphocytes, what happens? That is humoral immunity. Humoral immunity, which is possible by B lymphocytes. Here in the diagram, you can see a mature B cell having the antibodies on the surface to recognize the particular microbe, a free antigen who is moving in the body fluids, recognized by the antibodies which are present on the cell surface, a mature B cell having the antibodies to recognize that free antigens are moving in the body fluids. And here you see the MHC class 2 molecules on the cell surface. With the help of MHC class 2 molecules, they are submitting the antigen. The processed antigen is given to the T helper cells. Then further, the T helper cells again activates the B lymphocytes. Means earlier B lymphocyte acted as APC. When it was given to the T helper cell, the T helper cells activates the B lymphocytes again. Now it is going to participate in the humoral immunity by differentiating as plasma cells and memory cells. Memory cells for the further usage, and the plasma cells now started producing an antibody that which is specific to that particular microbe. So these are the antibodies which are produced by the plasma cell after the differentiation from the activation of the T helper cell. A mature B cell differentiated into two types of cells, plasma cell and memory cell, whereas plasma cell now producing antibodies, and in future the memory cells are going to produce the antibodies. In future, what I mean, when the microbe again enters into the body, that memory cells are going to be activated for that particular antigen, for that particular microbe, to produce the antibodies immediately. Then now the question raises, what happens? What these antibodies do? Antibodies which are produced from the plasma cells and released into the body fluids, what is the role of antibodies? To destroy that antigens who are moving in the body fluids. The produced antibodies, here you see that particular antibodies are attached to the virus in a specific place. We say that as antigenic fragment place of the microbe. Then they attach to that, they blocked that particular microbe, whether it is bacterium or virus. We call this process as neutralization. We say this process as neutralization. Then sometimes that antibodies, when they bind with that particular microbe, antigenic fragment of the microbe, they are going to form agglutination, clumping. In clumping form, you can see that particular antigen and antibody complex, and sometimes The antibody holds the microbe, antigenic fragment of the microbe, and that can be seen in the form of precipitation. So either the antibodies released from the B lymphocyte, the differentiated B lymphocytes, we call them as plasma cells, that antibodies will make either neutralization, they blocks the microbe, or they may form the clumping, agglutination, or they may form the precipitation. We say in general all the three aspects as antigen and antibody complex. This particular complex now engulfed by the phagocytes which are present in the blood cell. That means antibodies, when they bind and hold the microbe, it will facilitate our body phagocytes like macrophages will come and engulf and remove that particular the antigen and antibody complex from here. We call this process, we say it as humoral immunity. Humoral immunity. Some questions may raise here, but that we will discuss in acquired immunity in the part 4. We discuss that particular issues here. The basic process of humoral immunity is just like this. So finally, we have completed both humoral immunity and cell-mediated immunity. The basic issues to be remembered here, that B lymphocytes produced antibodies are participating in the immunity. We say such kind of immunity as humoral immunity, or T cytotoxic cells are responsible for the destruction of the microbe. We call that process as cell-mediated immunity. The basic difference between these two is, as you observed in the cell-mediated immunity, that antigens which are entered into the cell for that particular kind of microbes, that only possibility here, we say that kind of cell as altered cell, only possibility here the cytotoxic cell should release the perforins and grangenes and make the cell to be into pieces. The cell should be fragmented over, the cell should be, our body cell need to be destructed over. Such kind of possibility is only with T cytotoxic cells. That means here for endogenous antigens, that microbes which can enter into the cells, the best possibility is going to be T cytotoxic cells. You may have seen in the part 2 that cytotoxic cells also against that exogenous antigens, but more effective is for endogenous. Also they participate in the exogenous antigens. They are more effective against that endogenous antigens who are entered into the cells of our body. Then when it comes to the B lymphocyte, they release antibodies. When you see that these are very effective against that exogenous antigens, that means the microbes are roaming in the body fluids. So to find out the B lymphocytes differentiated in the form of plasma cells, and that plasma cells release antibodies. The released antibodies will recognize that antigen wherever it is, and they recognize and they form either precipitation or agglutination, or they neutralize the microbe for further phagocytosis. We call that process as humoral immunity. Now let's discuss about antibodies here. Antibodies. If you see a particular antibody, that any antibody is having 4 polypeptide chains. See the diagram? These are the 2 heavy polypeptide chains you can see, and these 2 are the light polypeptide chains. So any antibody is made up of 4 polypeptide chains, and which bond each other with disulfide bonds. And antibody is generally a protein. Any antibody is going to be protein. So basically there are 2 portions in the antibody. Fragment antigen binding site, we call FAB, or fragment constant region. Fragment antigen binding site, this is the place we can say fragment antigen binding site, this part, fragment antigen binding site, and this portion we say fragment constant region. The fragment antigen binding site is going to modify changes according to the antigenic fragment. based on that antigenic fragment, that fragment antigen binding site is going to be changed. This portion, the portion of antibody which binds with antigen can be considered as para 2. That can be considered as para 2. Now, let us see the fragment constant region. Basically, antibodies are of two types. Some antibodies will attach to the cell surfaces like B lymphocytes and macrophages, and earlier we have seen in the humoral immunity about the free antibodies which are moving in the body fluids. So two types of antibodies you can see generally, and based on their nature we have 5 types of antibody. We will discuss that in the coming parts. Whereas these particular antibodies, when they attach to the cell surfaces, we say fragment constant region. In general, the antibodies attaches to, with the help of FCN, they attaches to the B lymphocyte, and the next frequency you may see these mast cells, special kind of cells in our blood, that you can see that antibodies attaches there also. So B lymphocytes and mast cells is having that attached antibodies on their cell surfaces. Antibody attaches to them with the help of Fc. Then in general, antibodies are considered as immunoglobulins. Immunoglobulins. Briefly we say Ig. So these Ig immunoglobulins are generally are of like IgG, IgM, IgA, IgD and IgE. There are 5 types of antibodies: IgG, IgM, IgA, IgD and IgE. All these 5 types of antibodies will be discussed in the coming classes. Thank you, and please visit neetsrikanth.blogspot.com for the chapter wise question papers and consolidated tests, grand tests, and the useful videos which will make your studies easier. Thank you.